Welcome back to my channel Back and By World. Today I'll be discussing two important forms of chromatin which is found in the nucleus of a eukaryotic cell, that is euchromatin and heterochromatin. I'll be diving into the details of these both. And first of all, what is a chromatin? Basically, chromatin is a complex of DNA and proteins which results in the formation of chromosomes or that packages into the nucleus. Okay, it is a complex of DNA and proteins which helps in the formation of the chromosomes, and as a result of which packaging of the DNA can take place in a very small, we can say, micrometer nucleus. Okay, and this is basically a picture, and the we can say the differences in the staining of euchromatin and a heterochromatin. That is, if I stain a particular nucleus, okay, if I stain a nucleus, and these will be the you can say the result of it. Okay, there will be euchromatin is a lightly stained one, and whereas a heterochromatin, that is there are two forms of chromatin, euchromatin, euchromatin is a lightly stained part of the chromatin, whereas heterochromatin is the darkly, we can say, stained one. Okay, this is a nucleolus and this is the nuclear envelope. Okay, we all know that is nucleus double membrane, and basically this is the result of the staining if I stain a particular nucleus. Okay, and so this is basically a picture to which depicts the we can say euchromatin and the heterochromatin. Okay, as you can clearly see in the picture that euchromatin is that part of the chromatin which is usually loosely packed, you can say the packaging of it, whereas you can see here that is they're tightly packed. These are the nucleosomes. Okay, the DNA is packaged with the help of proteins which are termed as, we can say, protein complexes which are termed as histones, and the resultant structure is the nucleosomes. Okay, and the nucleosomes in case of the euchromatin, which is, it is not that tightly packed. Okay, and as a result of which this loosely packed, what happens, the DNA polymerases, or we can say the RNA polymerases, can come here and they can transcribe these part of the we can say the genome. Whereas in case of heterochromatin, they are very tightly packed. The nucleosomes are very tightly packed, and as a result of which these part of the chromatin is usually the silent one. Okay, by silent here it means that it rarely gets transcribed. Okay, the number of the, we can say the frequency, the rate at which the transcription of the DNA of the genome takes place is high in case of the euchromatin, whereas it is very rare that a particular gene present in the heterochromatin part of the genome is transcribed. Okay, I hope some things are clear from this, and it will be more clear in this slide. This is a detailed, we can say, the differences between the euchromatin and the heterochromatin parts of the, we can say, the chromatin, the we can say condensed form of our DNA. Condensation in case of the chromatin is less. I have already shown in the last slide. There is the condensation, that is the packaging of the nucleosome is very less, whereas in heterochromatin it is very more. It is densely packed, we can say. Okay, and staining, it is lightly stained, and whereas heterochromatin is usually darkly stained. Okay, now what is the location of the euchromatin and heterochromatin? Is there a difference? Do we have kind of, we can say, the landmarks where I can say that yes, euchromatin is present over here, heterochromatin is present over here? So location of this euchromatin is present in the chromosome arms. Okay, euchromatin is present in the chromosome arm, and whereas the heterochromatin is present in the centromere, or we can say telomere, and we can see some specific parts of a genome. We all know that the chromosome is in the shape of like this, we can say the 8. This is a centromere. Okay, the center one is a centromere, and the ends one are termed as telomeres. Okay, and the chromosome arm refer to these parts. Okay, whereas centromere, this is the centromere. Okay, this is the centromere, the center part, whereas these are the telomeres. Okay, I hope the location is clear. Where do we find euchromatin and heterochromatin? Next, types of the sequence. The sequences which are found in the euchromatin is unique. That is the A, T, G, C. The sequences are unique. Whereas in case of heterochromatin, the sequence is repeated. We can have 80, 80, 80, we can have GC, 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 like this. These are repeated. That is, the nucleotides are in repeated case. Okay, active genes, the number of the active genes. I have already told in the last slide that euchromatin is actively transcribed. Okay, and the number of the active genes are many in case of the euchromatin, whereas in case of heterochromatin, there are less number of genes which are active. Okay, next, total number of genes. There are two types of genes: active genes or total genes. Total genes is less in case of the euchromatin. They are less in case of the euchromatin and more in case of heterochromatin. But the number of the active genes are many in case of euchromatin and less in case of the heterochromatin. Okay, when does the replication of these parts occur? The euchromatin gets replicated in the early S phase, whereas in case of the heterochromatin, it gets it gets replicated at the late S phase. Okay, transcription here, it is frequent in case of the euchromatin part of the my genome, and whereas it is rare in case of the heterochromatin. Crossing over, that is a recombination, which usually occurs in case of the meiosis 1, and the crossing over is very common in case of the euchromatin. Okay, whereas it is rare in case of the heterochromatin. Now the sequences. Euchromatin is usually GC rich. That is a GC. That is, we can say, the GC rich sequences, GC rich genes are found in the euchromatin, whereas in case of the heterochromatin, the sequences are usually AT rich. Okay, I hope this is clear. Thank you so much for watching my channel. Please like, subscribe and share. Thank you.